Thank you again, and also thank you for all the participants who are going to join us today. I can start by saying that I have no financial disclosures. And also, I have to tell you that I'm going to present our work on sleep and activity patterns in ASD, but I do need to clarify that I'm not a physician, and my presentation today will not cover topics that are related to interventions, therapy, medication for sleep problems. However, going through the list of SPARK webinars, I found really impressive collection of presentations on relevant topic. So I would suggest you that for additional information about interventions that you visit or hear again presentations by Dr. Mello, Buckley, or Batts. Autism is highly complex. Impairments in social interaction and repetitive behavior represent the core ASD diagnostic criteria. But in addition to intellectual disability present in 30 to 40% of individuals with autism have several other, we call them ASD-linked traits and conditions, such as anxiety, epilepsy, anomalies or problems with executive function, sensory sensitivity, motor disturbances, and sleep disturbances that we are going to discuss today. So you may ask yourself, why are we scientists interested in these autism-linked traits and conditions? There are really several reasons. First of all, we are very interested in gaining mechanistic understanding for underlying impairments, and also relationship between the core features of autism and these common comorbidities. In an effort to gain mechanistic understanding, our studies in model organisms are very important. For example, mice or flies or even worms. Second reason is that we are very interested to improve lives of individuals with autism. And by understanding these ASD-linked traits and conditions, we may be able to alleviate or at least reduce some of these common comorbidities. And then finally, we are interested to identify target symptom domains that may be responsive to pharmacological treatment. So this pharmacological treatment can then, of course, improve lives. So another important question is, why do we study sleep? First of all, sleep problems are very common. And this is a theme that we will be revisiting over and over again in my presentation. Sleep often represents also an early or first sign in autism, and sleep plays a very important role in brain development. In terms of how common it is, I like to revisit SPARK data, specifically a paper that was published by SPARK Consortium with reports from caregivers who, according to their records, reported in approximately 40% of cases that ASD individual has sleep problems or sleep diagnosis, in comparison to approximately 10% in non-ASD individuals. It is also interesting that the rate of ASD problems is slightly higher in individuals with ASD with intellectual disability in comparison to individuals without intellectual disability. In my view, particularly telling are data that are coming from so-called quartet families, families where we have data on from caregivers reports on both parents and two children, one with autism and one unaffected sibling. You can see same number that we have seen before among probants and the individuals. The sleep problems are reported in 40%. On the other hand, this number is 10 or less percent in the father, mother, or unaffected sibling. Another very interesting aspect for us researchers is that it seems that there is a convergence of evidence that synaptic dysregulation plays very important role in autism and also in sleep disturbances. And this dysregulation can be studied not only in humans, but also in model organisms. So the big question is, how is our sleep or timing of sleep regulated? I'm going to share here with you amazing model, so-called two-process model of sleep, reported by Borbelli in early 90s, that still holds and leads and governs a lot of our studies. So according to this model, the timing of sleep-wake cycles is influenced by two highly interconnected fundamental biological processes: circadian process and homeostatic process. Circadian process controls daily rhythms in physiology and behavior, and the cycling persists even in constant darkness. So during these cycles, there is transition from circadian wake promotion to circadian sleep promotion. This period of these cycles is approximately 24 hours, and the name circadian comes from this approximately 24 hours, circadian approximately 24 hours. It is also very interesting that these cycles persist even under constant conditions. Homeostatic regulation of sleep mediates gradual rise in sleep pressure during wakefulness, and vice versa. The longer we sleep, there is a gradual rise in our readiness to wake up. We know a lot about genes that control circadian rhythms. And in fact, several years ago, Nobel Prize was given to researchers to study this process in fruit flies. Less is known about genes that control homeostatic process. However, based on studies in model organisms, we have a very comprehensive list of approximately 120 genes, which when mutated, lead to sleep and circadian disturbances in model organisms, and in some of them also validated in humans. And both of these processes, circadian process and homeostatic process, are require a proper synaptic function. So synapse development and function is extremely important for both sleep and for sleep-wake cycle. Based on genetic studies, we know that several risk genes in autism play a very important role in synapse development and function. And we also know from some very elegant neurobiological studies that there are changes in ultrastructure in many synapses during a sleep-wake rhythm. But there is also this very striking bidirectional relationship between sleep and ASD. Insufficient sleep exacerbates ASD symptoms and leads to poor social skills. And children with autism who get back on a regular sleeping schedule seem to learn better. They're less irritable and have fewer behavioral problems. However, the inverse is also true. Behavioral problems, for example, anxiety or social anxiety, can worsen sleep problems. So how do we monitor sleep? The best way, gold standard way, is to do it in the sleep lab, using multiple sensors, as illustrated here. These sensors can then detect the brain waves, brain activity, eye movement, muscle tone, heart rate, airflow sensors. But the major issue is that with polysomnography, we are getting really 1 day of data. We know that particularly in studies of sleep in autism, longitudinal data are very important. Not only that, but but children and adults with autism may have very hard time spending time in the sleep lab. My interest in sleep and autism really was influenced by a great collaboration with Dr. Margaret Souders, who is professor of nursing at Children's Hospital in Philadelphia. And it was more than 15 years ago that my lab helped her to analyze data that she collected with actimetry, accelerometer device. In fact, Margaret would give pajamas to a child, and child would spend several days or weeks getting acclimated to a new pajama. And the pajama had a small pocket where she would place accelerometer that I'm going to show you in the next slide. And her relatively small study provided very, very important preliminary data that monitoring activity just during night can show significant differences between ASD children or child and non-ASD sibling. So my laboratory explored several activity devices. The first one, Philips Respironics, was originally used, and it's broadly used in sleep clinics. We compared several research devices such as Gene Active and Activity. On the other hand, as you all know, there are many consumer devices such as Fitbit and Aura. So for research purposes, we prefer to use a research device because we have complete control of data. We know how these data are collected and analyzed. With Fitbit and Aura, we can get data. However, all the algorithms that I use are proprietary. So for us, this is a black box, how they're deriving different activity and sleep traits. In the case of Aura, they're actually providing some data, but the resolution of this data is not as good as resolution that we get using our research devices. So here I'm illustrating for you visualization of our data. So this is so-called, we call it actigraph. It's double plotted record because each day, as you can see here and here, or another day here and here, each day is plotted 2 times so that we have a nice overview of both activity and rest phase. In yellow is a photo period, time of the day, daylight, and we actually use time or season when recording was done, and also longitude and latitude to precisely determine this photo period. So you can see here, these are periods of activity and then much, much less movement that's detected during the night. So using our algorithms, we can now go and extract large number, over 200 different measures. So, and these measures and analysis is very different for activity, and this is a reason for title of my talk. It's not only sleep, but also activity. So we measure level of activity and onset of N10 phase as a measure of overall daytime activity, and then L5, the least active 5 hours during the night. Using L5 and M10, we can then calculate many different aspects, many different traits. To study sleep quality, we evaluate wake after sleep onset and sleep efficiency. And relative amplitude is the difference between average nighttime restlessness and average daytime activity. Relative amplitude is particularly important because it reflects the strength of circadian rhythms. So this is our 3-week record. I'm also going to show you here just 1 day of recording, and here you can see this 1 day presented from midnight to midnight, or from noon to noon. And you can see now here times of very, very high level of activity, times with lower level of activity that indicates sleep onset. Use our algorithms now integrate all this data and then give us precise overview of times when person is very likely asleep and times when they are awake during that entire sleep period. In addition to activity, in addition to the angle of sensor that give us a position of the arm and arm movement, we also record temperature and light. And this is, of course, near body temperature. And these are measures that are very helpful for us when we do and perform quality control of our data to see, and also you'll be able to see how many individuals that are included in our study are actually having light entire night that can have very important effect on their rest activity pattern. So the next important step is analysis of data. We use several algorithms for the analysis. Some of these algorithms, like for instance, Gene-active and gene data in R, it gives us a wide range of both activity and sleep traits. This is now commonly used algorithm and has been used, for instance, for the analysis of thousands and thousands of records for individuals who participate in UK Biobank. Another algorithm called Chronosapiens is particularly useful for the analysis of circadian traits. Rest activity pattern or data are fitted to sinusoid curve, and there are basically traits that can indicate, of course, a relative amplitude and several other circadian traits. As you will see, our preliminary data show very striking changes and effects on physical activity. So we are now using an algorithm called accelerometer that basically provides classification of different level of activity. It can distinguish sitting from low level activity, and then also identify periods of very high rigorous activity. So the big question in our study is the heritability of these traits. So we have so far analyzed actimetry in over 1,000 individuals, but our initial study started in twins. And this data showed us that sleep and rest activity traits have moderate heritability between 40 and 60%. So it means that genes play a very important role, but we cannot ignore environmental component. And that's also very important when we think about interventions and treatment. So I'm going to give you just a relatively brief progress on our current studies. Our studies are ongoing. It is absolutely clear that the number of individuals that will have to be analyzed is high, and for that reason, this is just a progress report. So we started several years ago by recruiting extended families from individuals with autism in our ASPI program at Penn, and we have actimetry data on 150 ASD subjects and approximately 230 family members. And our study supported by Simon's Foundation on SPARK individuals consented so far 700 individuals. And for 700 individuals, we have also very extensive questionnaire data. And so far, we have completed actimetry analysis, and we got high quality actimetry data for 200 subjects. So altogether, to our knowledge, this is the largest study, actimetry study in autism. So you may ask, particularly families who participated already in who enrolled in our study, why are we going through all these questionnaires? So we are basically using social responsiveness scale to learn more about ASD traits, as well as broad autism phenotype questionnaire. And then we are using brief to be able to learn more about executive function. We have several questionnaires that give us some information about sleep disturbances and sleep impairment, daytime sleepiness. And finally, we also included in SPARK study, unfortunately, we don't have this data in ASPI, so-called Glasgow sensitivity questionnaire that gives us some information about sensory sensitivity. So all together, for each individual, we have approximately 250 measures, and we are using very sophisticated machine learning methods to prioritize traits and identify traits that are particularly relevant to autism. But going back to that question, why do we need all these questionnaires? I will go back to the slide that I showed you before. On the long run, we would like to learn more about a relationship between social impairment and sleep disturbances and anxiety and executive function. And basically, in order to give you some sense for the richness of data that we are generating, I'm going to present preliminary scores from Glasgow sensory questionnaire that was already done on almost 700 subjects. So you can see here a frequency distribution of all 700 individuals according to published data. The mean is somewhere around slightly higher than 50, and you can see a shift in individual in SPARK probants, in SPARK ASD individual. As you will see later on, we do not have controls. We are about to launch analysis and enrollment of controls, but so far as controls, we are using unaffected relatives from ASPI probants, ASPI individuals. So as you can see here, there is a significant number of individuals who have very, very low GSQ scores, do not report major sensory sensitivity issues. And then individuals here at the end with pronounced sensitivity scores, high sensitivity scores. Again, just to give you a flavor for data that we're getting so far, we are getting a Glasgow sensitivity questionnaire. It gives a score for different sensory modalities, and it is from auditory, gustatory, olfactory, and so on. With a very striking, interesting result, but not surprising for many of our colleagues, is that particularly high scores, it means high hypersensitivity and high hyposensitivity is detected for auditory sensitivity. So going back to sleep, here I'm showing distribution of scores based on promise. As we have seen before, there is significant number of individuals who do not report sleep disturbances. In our study, we are using informant information because it's a study is designed or started in such a way that individuals should be able to fill in questionnaires. And then you can see also the significant difference between mean score that's reported in the literature and also mean score for this group of 700 individuals with autism. So going back to this slide here, the interesting thing is that to look at these activity records for individuals who have profound autism, who have very, very high scores, for instance, on social responsiveness scale. They have, on the other hand, in many cases, their activity records are absolutely perfect. Actually, in some cases, these records, the level of, there is a great consistency in the pattern of activity from one individual to the other. On the other hand, when we go to the other end, and look at the patterns of individuals with very, very high promise scores, you can see individuals like this one here with extremely low level of activity during the day and highly fragmented sleep during the night. Another irregular record with high level of activity, very little sleep, very irregular sleep onset, sleep or activity onset. And then I'm showing you here another striking activity record, and it is very interesting that we actually have several individuals who have this pattern that when we have double plotted activity record plotted to 48 hours, it looks like the individual is waking up every day later and later. These activity records are very particularly striking for individuals who are involved or interested in circadian behavior, because these individuals seem as individuals who are not entrained and who are not actually responsive to environmental cues, because our circadian system is entrained by getting, for instance, light cues or food cues from food and so on. On the other hand, it will be very important to know whether this individual had very unusual time or whether this same record will be detected using longitudinal recording. I am comparing these records to a striking report published almost 20 years ago by Japanese neurologist and scientist, Dr. Segawa, who reported in this particular book chapter, a year-long record for a child whose mother reported a sleep pattern. And this child had these episodic times when it looked like the child is not responsive to environmental cues. So for individuals that with similar pattern that we detected, it would be very important to go back and try to perform longitudinal recording. So I mentioned to you that we're using machine learning methods to prioritize the important sleep and activity traits, but here I'm showing conventionally generated graphs just to give you some sense for type of differences that we are detecting between ASPI relatives who do not have autism and individuals with autism from SPARC and from ASPI. So, and there are two categories of traits that I want to illustrate to you here. First of all, delayed sleep onset in individuals with autism in comparison to unaffected individuals who do not have autism, and corresponding to a late sleep onset, also a later wake time. However, we have also detected that as significant traits, several traits related to physical activity, specifically in individuals with autism, we detected lower level of physical activity and higher duration of inactivity bouts of 30 minutes. With all this, I can also mention that the major advantage of performing this type of studies at the time when we have also genomic information is that we can look for specific genetic profile for all these individuals. There are very sophisticated genetic methods that allow us to look at so-called polygenic risk score, but I'm illustrating here only a first step. Basically, when I mentioned to you two-process model of sleep, regulation of sleep-wake cycles, I mentioned that we have this impressive list of 120 genes that are involved in circadian and sleep regulation. So we can turn to our ASPI whole genome sequence and also to genomic information for SPARC and identify mutations in several genes involved in regulation of circadian rhythms or genes that are known to be linked to sleep disturbances in model organisms. And for aficionados and those who are more familiar with genomic analysis, I'm listing here criteria for our report of this rare, or actually, in fact, moderately rare predicted deleterious variants in the sleep and clock genes. Of course, as I mentioned earlier, many of these findings will, of course, have to be repeated because mutations that we are generating we're detecting in humans are heterozygous, so there is only one copy of that variant found in the individual. So I'm now going to summarize our findings to date. Actimetry can be conducted over prolonged periods, enabling assessment of sleep dynamics and activity on a scale that's sufficient for genetic investigations. Of course, we would like to have even higher scale than what we're doing right now. Our major findings are that autism individuals, probants, show lower level of physical activity during the day and very high degree of variability across both sleep and activity traits. And integration of human genetics findings in these ASD families with parallel studies in model organisms will be really necessary to understand the role of the synaptic imbalance and the basis for ASD associated sleep problems. So this is ongoing study. It is extremely important to continue and to be able to enroll and monitor activity in large number of individuals with autism and ideally unaffected family members. But our main challenges is genetic heterogeneity. I showed you, for instance, mutations in many of known sleep and clock genes. These mutations are found in very, very rare, and they're found only in small number of individuals. There is high degree of day-to-day variability in sleep and activity patterns. And this is one reason why it will be very, very useful and helpful to have longitudinal reporting, not just 3 weeks, but actually much longer. There are significant sex differences in sleep and activity patterns. And we have noticed these sex differences, particularly when we look at larger number of individuals with mutations, with pathogenic variants in specific genes. There are significant age differences in sleep and activity patterns. So this is one reason why it would be important to have this longitudinal sleep activity monitoring. And then finally, research device that we are using right now detects angle, activity, temperature, and light. It would be important to combine that with additional data such as heart rate variability, oxygen saturation, and more importantly, EEG, electrical activity. And this is a great opportunity for me to present a very, very exciting project that is going to be launched, I learned next week, Simon's pilot project directed by Dr. Ilan Dinstein, who is actually going to join us today for a panel when you will have a chance to ask us questions. So this study is going to use 2 devices, Embrace Plus and a Dream, that will be able to actually detect EEG scores. And the pilot will include analysis of 100 families with ASD, child to adolescent, and non-ASD sibling, and recording will be performed over 2 to 3 weeks. So looking ahead, in my view, one day sleep and activity patterns collected by wearable device will be an actionable part of clinical care of an individual with ASD. There is a lot of discussion about that around Penn and Children's Hospital. On the other hand, amount of data that we are getting from sensors is vast, and it will be extremely important to turn to computer and data scientists and who can help us to integrate this data and learn more about our biology and interaction between our biology and environment. So at the end, I would like to acknowledge and thank both ASPI and SPARC participants. I went through all the questionnaires that we share with you, and I'm incredibly grateful for patience and high quality of data that we are getting in our studies. I would like to thank members of my laboratory, my collaborators in the ASPI, and also collaborators who are helping us with analysis. Today, with me are Dr. Ted Brodkin, who is physician scientist, psychiatrist, working on autism, Matt Kaiser, also physician scientist and sleep clinician, and Ilan Dinstein, whom I mentioned already, who is launching a new SPAR study. So with this, I would like to thank you for your attention, and we will switch to questions.
Thank you so much, Dr. Bućan. Yeah, and it's a pleasure to have Dr. Brodkin, Kaiser, and Dinstein join us today for this Q&A portion. So let's just jump right in because we only have about 15 minutes, and I'm hopeful that everyone that's on this panel now will get a chance to help answer some of our attendee questions. So starting with questions directly related to what you just presented, Dr. Bućan. So in your findings, you indicated that ASD individuals showed lower physical activity levels. Was there any sense of what high physical activity levels may have showed in this study for individuals with ASD?
So we are still analyzing data, and we know that there are differences in different categories of physical activity. There are differences in, sedentary activity. There are differences in vigorous activity. We are still analyzing that.
Okay, great.
So it's actually very interesting. We are able to use, I mentioned that we're using accelerometer method. So it's basically a study where 300 individuals had cameras for 3 weeks. So they had activity device and camera, and using machine learning, a program learned how is, for instance, driving a bicycle different than sitting in the chair and so on. So these are very, very sophisticated methods, but interpretation of kind of physical activity that we detect is still, we're still working on it.
Great, thank you. And then through this study, this was kind of an interesting question that came in, more about just general observations. Have you noticed any specific, with these children with autism, any specific physical behaviors that they're exuding while they're sleeping? Like, for instance, sleeping with their mouth open, that just as an example.
And unfortunately, we can't say that because we don't have this type of recording. But I think that Matt may be able to address that, because Matt, many of your patients are, even if they don't have autism, but maybe you had some with autism. These are kind of observations that you can make in sleep lab, of course. Mm-hmm. Matt, can you address that?
Yeah, I think that's a really interesting question. I saw a number of questions related to how primary sleep disorders like restless legs, or someone, it seems, I think, wrote about periodic limb movement disorder may be involved in the sleep disturbance in autism. I think that that's an interesting area that's probably underexplored, I think, and probably under-recognized. And certainly things like restless legs and periodic limb movements are well known to be associated with low iron levels, for example. And so dietary preferences among people with autism spectrum disorder may secondarily lead to limb movement disorders as it relates to iron, for example. But I think from what we know, that's not the predominant cause, which is to say that the sleep disturbances in autism that are heterogeneous but widely observed can't uniformly be related to a primary sleep disorder in most cases, like restless legs or like periodic limb movement disorder or something else like that, as opposed as Dr. Bućan already said, to primary issues related to how the brain maybe has developed or how sleep circuits have developed, or something like that. These are really interesting and important questions, though. Perhaps Dr. Brodkin has more to say since he treats people with autism more often than I do. I'm not sure, though, on the sleep side.
I think you summarized it really well, Matt. I don't have much to add to that.
Like sort of, I guess, in the similar vein of observing sleep patterns. So, many children and adults with autism do have a struggle with some sensory sensitivities. So for individuals that may have hypersensitivity to wearing any of these devices, do you find that there's any benefit or any to observing sleep patterns, just as it sounds like Dr. Kaiser and Dr. Broken, you may do in your labs as a form of a treatment, or to indicate what treatment would be necessary?
You want to take that, Dr. Kaiser?
Yeah, I think something to point out is that the ability to do detailed sleep studies, that's going to be something that needs to be approved by an insurance company. It requires an overnight stay in the hospital or in a hotel, I should say, usually is where these are done. So that's not going to be a standard of care unless there's something on the history to lead a physician, a treating physician, to suspect that there's something is going on. And in fact, nowadays for sleep apnea, for example, the first line is usually assessed with a home sleep study where something is sent to someone's home as opposed to it being done. So then you don't even get all this extra information that you're talking about right now. Dr. Bućan would be more aware than I am of any attempts, although relatively low throughput, to do detailed studies of individuals with autism to address some of these questions. But I think it's really important to emphasize what Dr. Bućan was trying to, has so nicely laid out, which is there's probably a lot of different forms of sleep problems, just like there are a lot of different causes of autism. So looking one by one is not really going to help us shed light on sort of broader principles that might be at play here. I do think it's pretty clear that a primary sleep disorder, let's just take restless legs, okay, as an example, is not across the board a common cause of sleep problems in autism. Does that make sense? It could be in some cases, but it's not usually going to be what's at play.
So we are really going very carefully, record by record, and it's so interesting to see sleep apnea. These are individuals who are diagnosed with sleep apnea. And in some cases, you see highly irregular sleep pattern. And then in some cases, you don't see using actimetry. So of course, in situations like that, it would be very important to be in the sleep lab, or in my view, I'm incredibly impressed by Simon's pilot study, because the dream bands could be extremely important in situations like that.
Can I add just a quick addition?
Please.
So I think that there have been quite a lot of studies showing a relationship between sensory sensitivities and sleep problems, and it's actually the strongest relationship of any symptom with sleep problems that you see in autistic kids. So like in the same way that there's heterogeneity in sleep problems, there's heterogeneity in the different kind of sensory problems that that autistic individuals have, and so mapping that right is challenging. In certain individuals, those things go together, and others they will not. And the other there was another question about relationship with other maybe symptoms of autistic individuals. Another really big one is irritability and kind of tantrums during the day and things like that go together with these sleep disturbances. There have been several studies showing that.
Mm-hmm. So I'm incredibly impressed by questions. We could spend hours going and addressing these questions. And I want to make sure, Dominic, that we will have a way of going back and addressing, we can't do it in next 7 minutes.
Yeah, absolutely.
But we
Yes.
have a way of being able to go back, and also we will definitely provide information, or you can provide information through Spark on our ongoing Spark study.
Yeah, absolutely. So we've collected, or we are actively, as we speak, collecting the questions that have come in, so we can share those with everyone on this panel. So anything that we can't get to, if any of you want to expand upon that, you can share that with me, and then we can make it available along with this recording and the slides on Spark site. Thank you so much. But while we do still have a few minutes, let's hope maybe we can get to one or two more. I think this has been a great conversation. So going back to some of the questionnaires that you use and the data you collect before people begin participating in the study, can you talk about if you're collecting data on different comorbidities in these individuals with autism, such as, ADHD is one that, people ask about a lot?
Yes, so we collect, but many of these data were actually already available in Spark from caregivers' report. On the other hand, it will be very interesting to compare caregivers' report with report, because in some cases, this is going to be several years after. Do we have individuals who did not have sleep problems based on caregivers' reports, but now there is a self-report that there are problems. So these longitudinal data are going to be incredibly valuable.
I also noticed in the chat, someone asked how many kids on the autism spectrum also have an ADHD diagnosis.
Yeah.
Speaking of comorbidity, and I think it's roughly 50% or so, 50 or maybe even 60%. At some point, we'll get an ADHD diagnosis. Sorry if I interrupted you, Dr.
No, that's fine. And I would ask my co-panelists to look at the question, and then you can pick question that you want to address. And I am going to try to address Aline said, wild question, wonder whether Spark will allow people to link Apple Watch Fitbit for people with autism to send actimetry data to researchers. So this is a great question. I think that would be perfect. A few days ago at University of Pennsylvania, we had a mother who shared with all of us that she had Fitbit data for her child when she went to the sleep clinic. And right now there is no way, and I don't know whether it's no way or no expertise to use this data. On the other hand, exploring Apple Watch data and Fitbit would be really helpful. As I said, it's not high resolution. We don't know how this data are generated, but it will be still very helpful. So, Ilan, is there a question that you would like to address? I'll ask each one of you to pick a question.
I saw that several participants were asking about the relationship with physical activity, and could actually enhanced physical activity be part of what's causing maybe some sleep problems. And this is where, again, the heterogeneity comes in, right? So different individuals have different baseline levels of activity. Some of us are more active than others, used to different levels of physical activity. Part of the reason why we're launching this next study at the foundation is to look at within individual participants, to what degree does their night sleep correspond to the level of activity in the previous day. And it might not be just a matter of just one day. You might have to look at the entire week and maybe compare a week where someone was more active versus a week where they were less active, and try and figure out how that perhaps contributes to sleep problems within an individual. But that's definitely a super interesting direction with very practical recommendations for maybe individual people.
So Matt, how about you? Do you have your favorite question?
Well, I saw that there were many questions about use of melatonin and other medications, which I can't give specific advice to individuals, unfortunately. But what I would emphasize, number one, is these are really important questions to be talking to your providers about. And number two, although I know resources with regard to treatment providers can be limited, for adults in general and for kids in general, not necessarily limited to folks with autism, behavioral interventions are the first line treatment for sleep challenges. And so I think, I know that that's sort of throwing off into the abyss because it's hard to find people with the right expertise. But I think that a really hard look at sleep hygiene, sleep patterns of the family, optimizing time spent in bed and things like that are critical towards helping those medications have as most robust a benefit as possible, because no medicines really work very well alone for insomnia, sleep troubles in a vacuum.
Mhm.
Now,
Can
just
I,
to- yeah, go ahead.
before you go, Ted, I will just say there is a question whether adults are included, both in Spark and ASPE, we're including adults.
That was gonna be my favorite question, so you anticipated, yes, that's
Yeah.
true.
But do you have any other favorite question, Ted?
I don't know, that was the one that stood out to me, but I think,
Yes.
yeah, I think Elon and Dr. Dinstein and Dr. Kaiser really picked up on the other major theme, so.
Well,